What's going on, guys? It's your boy Josh with Rise Above Health. I'm back again, this time with part 2 of the melatonin series. This video will cover the possible biohacking aspects of melatonin consumption, including effects on oxidative stress, aging, cancer treatment, and neuroprotection based on the latest medical literature available. Before I get started, please subscribe to the channel, like the video, and afterwards leave a comment if you find anything of interest or have any questions. First, I will briefly recap a summary of the previous melatonin video for introduction and then move into this one. Melatonin is considered the first-line pharmacologic therapy for the treatment of insomnia by the American Academy of Family Physicians. There are other indicators as well surrounding sleep-related concerns, including shift work and jet lag. However, the FDA does not regulate supplements, as supplements are generally considered safe. Melatonin is involved in regulating the body's sleep-wake cycles through its interactions with the suprachiasmatic nucleus, promoting sleep and inhibiting wake-promoting signals via interactions with the MT1 and MT2 receptors. It is made and released by the pineal gland, but also in the gut and mitochondria. Most studies that have been conducted for supplementary melatonin are generally in a range of up to 10 milligrams for 6 months or less. For the full review, or part 1, which contains studies on dosing of melatonin, please check out the link in the description of the video. I'll also include a card at the end so that you guys can find the link. Now, let's start the video off with some papers regarding the effects of melatonin on aging. From a study entitled Integration of Melatonin-Related Redox, Homeostasis, Aging, and Circadian Rhythm, quote, melatonin levels tend to decrease significantly with age. Recent reports show that disruptions of circadian rhythms may render aging populations even more susceptible to age-related disorders. Aging is a progressive and degenerative process in life. Although more than 300 theories are proposed to explain underlying mechanisms of aging and aspects related with this process, we still do not know how or why we age due to the innate complexity of the matter. Mitochondrial aging theories is one of the widely accepted ones. A common feature of aging is altered sleep patterns, and commonly the elderly start sleeping earlier than desired. Quote-unquote healthy older individuals show characteristic alterations in sleep patterns. This finding suggests that even though they are quote healthy individuals, their circadian rhythm homeostasis is misaligned. It could therefore be speculated that since melatonin synthesis decreases with aging, decreased melatonin levels could be one of the contributing factors in the development of age-related degenerative processes. Next, a paper entitled Melatonin and the Theories of Aging, a critical appraise of melatonin's role in anti-aging mechanisms. Several theories have tried to explain the mechanisms of aging and the limits of lifespan. It seems important to perceive the differences between these two aspects. In the relationship between melatonin and human aging, a crucial observation is the decline of the nocturnal melatonin peak in elderly persons. This is not equally pronounced in every individual, but clearly demonstrable on a statistical basis. On this background, melatonin has been hypothesized to prolong lifespan, which cannot be easily tested in humans. In laboratory mammals, the evidence for this assumption is relatively poor as well. Next, an older review from 1996 entitled Melatonin is an Anti-Aging Drug, Between Facts and Fantasy. The production of melatonin declines with increasing age, and the circadian melatonin levels are affected by certain pharmacological or physiological conditions and manipulations. Clinical studies need to be performed in order to identify possible side effects of long-term melatonin treatment, especially in elderly and diseased subjects, unquote. From these papers, we can see that melatonin has possible, if not anti-aging properties, but healthy aging promoting properties. As people age, they tend to produce less melatonin, so they may sleep less, and with lower sleep efficiency, have a raised possibility of continued low-grade systemic inflammation called inflammaging due to this. The available literature stresses that further research is needed. Next are the available review papers I found regarding the possible cancer-preventing or treatment-related properties of melatonin. Could it be helpful in treating certain types of cancers? Before we begin this section, it is very important to note that if you do have cancer, you should discuss what I am reviewing in this video with your doctor before making any changes whatsoever to your medication and supplement regimen. That is very important. I have decided not to cover the specifics of melatonin on different types of cancer. As is the case with cancer, the survivability of different cancer cells is tissue and context dependent, and so I recommend you research this in the context of whatever cancer you may have interest in looking up. Our understanding of cancer is still evolving, and the mechanisms that cause and fuel it are still being fully elucidated. First, a review entitled Anti-Warburg Effect of Melatonin. Quote, altered mitochondrial metabolism is referred to as aerobic glycolysis, or the Warburg effect, and is common in solid tumors and in other pathological cells. Switching from mitochondrial oxidative phosphorylation to aerobic glycolysis, molecules called glycolytics inhibit aerobic glycolysis and convert cells to a healthier phenotype. Melatonin is a glycolytic into that healthier phenotype. From a 2022 review elaborating on this, entitled Melatonin Inhibits Warburg-Dependent Cancer, quote, melatonin has the ability to intervene in the initiation, progression, and metastasis of some experimental cancers. Melatonin halts the proliferative activity of cancer cells, reducing their metastatic potential and causing them to more readily undergo apoptosis. Whereas melatonin is synthesized in the mitochondria of normal cells, we hypothesize this synthetic capability is not present in cancer cell mitochondria because of the depressed acetyl-CoA. Acetyl-CoA is necessary for the rate-limiting enzyme in melatonin synthesis. Finally, the ability of melatonin to switch glucose oxidation from the cytosol to mitochondria also explains how tumors that become resistant to conventional chemotherapies are resensitized to the same treatment when melatonin is applied. Unquote. Next, a look in on the effects of melatonin on the immune system entitled Boosting Immune System Against Cancer by Melatonin, a Mechanistic Viewpoint. Quote, cancer is a disease of high complexity. Resistance to therapies is a major challenge in cancer-targeted therapies. Natural killer cells and cytotoxic T lymphocytes are the main anti-cancer immune cells, while T regulatory cells, or Tregs, and cancer-associated fibroblasts, or CAFs, facilitate immune escape of cancer cells. Immune escape is when the host's immune system is no longer able to recognize a pathogen and eliminate it. Melatonin is a natural agent with anti-cancer functions that has also been suggested in combination with cancer therapy modalities such as chemotherapy, radiotherapy, immunotherapy, and tumor vaccination. One of the main effects of melatonin is the regulation of immune responses against cancer cells. Melatonin has been shown to potentiate the activities of anti-cancer immune cells, as well as attenuating the activities of Tregs and CAFs. It also has a potent effect on the mitochondria, which may change immune responses against cancer. And finally, one more review of the effects of melatonin on human trophoblasts, a pseudo-tumor promoting cell type. In a 2012 review, Melatonin, the Smart Killer. Quote, melatonin has the ability to induce intrinsic apoptosis in tumor cells while it inhibits it in non-tumor cells. Melatonin kills tumor cells through the induction of reactive oxygen species generation and activation of pro-apoptotic pathways. In contrast, melatonin promotes the survival of non-tumor cells due to its antioxidant properties and the inhibition of pro-apoptotic pathways. These findings suggest that the trophoblast is a good model to study the different effects of melatonin on the intrinsic apoptosis pathway. So there's good promise here for melatonin as an add-on for regulation of both healthy and cancerous cells, as it plays a very important role in regulating cellular metabolism, both normal and abnormal. And in the case of cancer cells, it appears to, in the studies that have been done, trend cells away from aerobic glycolysis to a more anaerobic activity. Now for neuroprotection, a review of the literature for neuroprotection in general terms, plus some human and rat models. First, a review paper entitled The Neuroprotective Role of Melatonin in Neurological Disorders. Melatonin is a neurohormone secreted from the pineal gland and has a wide range of regulatory and neuroprotective mechanisms. It is reported that melatonin levels is disturbed in some conditions such as stroke, Alzheimer's disease, and Parkinson's disease, which indicates its involvement in the pathophysiology of these diseases. Although the brain makes up only 2% of human body weight, it consumes around 20% of the body's oxygen. This high level of oxygen consumption can initiate a harmful process known as oxidative stress. Oxidative stress is the occurrence of reactive oxygen species in a way that exceeds the capacity of antioxidant effects. In models of Alzheimer's, it has been found that long-term application of melatonin for around 2 months decreased degenerative changes in the hippocampus, prevented mitochondrial dysfunction, and delayed anxiety and cognitive impairment in a sporadic rat model of Alzheimer's. Melatonin is also reported to be a potent antioxidant in Alzheimer's disease. Several studies have found that melatonin reduces amyloid beta-mediated oxidative stress and lipid peroxidase. It has been found that long-term administration of oral melatonin enhances hippocampal synaptic growth and preserves the neuronal and glial structure in a sporadic model of Alzheimer's. In Parkinson's, evidence has been accumulating that age-associated Parkinson's is combined with oxidative stress. By downregulating the oxidative stress effects, melatonin is shown to be a potent antioxidant which can improve prognosis in Parkinson's disease. Next, a paper entitled Melatonin's Neuroprotective Role in Mitochondria. Quote, "For decades now, melatonin has been implicated in promoting and maintaining sleep. More recently, evidence indicates that it also plays an important role in neuroprotection. There is ample evidence to support melatonin's role in neuroprotection. In fact, animal studies have found that melatonin is effective in scavenging free radicals during post-ischemic reperfusion and after head trauma. It should be noted that the time of melatonin administration is critical when treating head trauma. Melatonin reduces malondialdehyde, a marker of oxidative stress, when melatonin was administered within the first 2 hours post-trauma. If given 8 or even 48 hours after the injury has occurred, the melatonin only then increases malondialdehyde levels, though the reasons why are unclear. In addition to its antioxidant activity, melatonin promotes activities of antioxidant enzymes and reduces pro-oxidant enzymes. Now let's look at some other mouse, rat, and human models demonstrating neuroprotective effects of melatonin supplementation. In a 2018 study entitled Melatonin Induces Mechanisms of Brain Resilience Against Neurodegeneration, quote, "The results of the study demonstrate that in a transgenic mouse model, melatonin treatment protects against dementia behavior and promotes cognitive performance after 6 months of melatonin treatment. There was a protective effect against Alzheimer's disease-like pathology, underlying decreased dementia-like behavioral alterations, and enhanced learning and memory capabilities in 12-month-old mice. Strikingly, melatonin chronic administration also improved recognition memory and increased exploration and decreased anxiety traits in non-transgenic mice, demonstrating an effect of cognitive enhancement and well-being inducement. Melatonin administration decreased amyloid and phosphorylated tau burden in the hippocampus of 3 times transgenic Alzheimer's disease mice." Next, the human models against cognitive impairment. Melatonin for cognitive impairment, a systematic review, quote, "All relevant randomized controlled trials in which orally administered melatonin in any dosage was compared with a control group for the effect on managing cognitive, behavioral, and/or affective disturbances of people with dementia of any degree of severity. These 3 studies met the inclusion criteria. Individual study estimates for treatment effect demonstrated a significant improvement for melatonin compared with placebo in behavioral and affective symptoms as measured by the ADAS non-cognitive scale in a study of 20 patients, and the neuropsychiatric inventory following the treatment of 2.5 milligrams per day of standard release melatonin. The remainder of the treatments for affect, behavior, and activities of daily living were non-significant. Now, a systematic review and comparison of the effects of melatonin treatment in patients with Alzheimer's, entitled Neurocognitive Effects of Melatonin Treatment. 22 studies met the inclusion criteria. Alzheimer's patients receiving 12 or greater weeks of melatonin treatment improved mini mental state examination score. Importantly, melatonin significantly improved the MMSE score in a mild stage of Alzheimer's disease. Meta-analysis of the MMSE score suggested that melatonin is effective for the treatment of mild-stage Alzheimer's disease. Finally, we have a review of the effects of melatonin on oxidative stress. Oxidative stress is an overproduction accumulation of reactive oxygen species. They are normal and necessary byproducts of various cellular processes in the body, such as the production of ATP, but in excess can lead to cellular damage and decay, as well as damage to DNA, cardiovascular disease, neurological disease, and several others, while speeding up the aging process. This is reiterated here in a 2015 review paper entitled Protective Effects of Melatonin on Mitochondria-Targeted Antioxidants. Quote, melatonin and its metabolites are powerful antioxidants and scavengers of free radicals, protecting cellular membranes, the electron transporter chain, and mitochondrial DNA from oxidative damage. These properties may make melatonin a potent protector against a variety of free radical-related diseases. By comparison, other conventional antioxidants have less efficacy due to their limited access to the mitochondria. Mitochondria-targeted antioxidants accumulate in several hundredfold greater concentrations within mitochondria and protect these critical organelles from oxidative damage. Melatonin also seems to be a mitochondria-targeted antioxidant and has similar protective actions as synthetic antioxidants. From a 2020 entitled Effects of Melatonin Supplementation on Oxidative Stress Blood Marker Parameters, but a mouthful. A total of 12 randomized control trials were eligible. The meta-analysis indicated an association between melatonin intake and a significant increase in total antioxidant capacity, glutathione levels, superoxide dismutase, which breaks down reactive oxygen species, glutathione peroxidase, and glutathione reductase activities, and a significant reduction in malondialdehyde levels. The authors concluded that melatonin intake was shown to have a significant impact on improving oxidative stress parameters. Additionally, there was a considerable effect on total antioxidant levels by using 20 milligrams per day or more melatonin in people under 35 years old. Malondialdehyde levels also decreased using dosage of below 20 milligrams per day over 35 years old. In a human model of obesity, melatonin was seen to lower oxidative stress in obese patients on a calorie-restricted diet. In a 2021 study, quote, melatonin deficiency has been demonstrated to correlate with obesity. 30 obese patients were supplemented with a daily dose of 10 milligrams of melatonin, a group of 15 people, or placebo, a group of another 15 people, for 30 days with a calorie-restricted diet. Significant body weight reduction was observed only in the melatonin group. The results show an evidence of increased oxidative stress accompanying calorie restriction. Melatonin supplementation facilitated body weight reduction, improved antioxidant defense, and regulated adipokine secretion. The findings strongly suggest that melatonin should be considered in obesity management. Similarly, there was another randomized controlled trial in 2015 on the effects of melatonin in oxidative stress inflammation in obese women. In a randomized double-blind placebo-controlled trial, 44 obese women were randomly assigned to melatonin, 22 women, and placebo, 22 women. Subjects were supplemented with a daily dose of 6 milligrams of melatonin or placebo for a low-calorie diet for 40 days. In the melatonin group, mean tumor necrosis factor alpha, IL-6, high-sensitivity C-reactive protein, and MDA levels decreased significantly, while in the placebo group, the decrease in values were not significant. Mean total antioxidant level increased slightly in the melatonin group, whereas it decreased slightly in the placebo group. Significant differences were observed only for TNFA and IL-6 between the two study groups. In conclusion, melatonin and melatonin supplementation in particular have a plethora of potentially healthful effects in the body, including promotion of normal and healthy sleep-wake cycles, healthful aging by way of circadian rhythm, and redox homeostasis. Melatonin may also be helpful in resensitizing cancer patients to treatment by reversal of the Warburg effect, but please discuss this with your doctor. Melatonin has been shown in the studies to potentiate the activity of anti-cancer immune cells, as well as attenuating the activities of Treg cells and CAFs, which would normally help cancer cells to evade the immune system. Melatonin also has neuroprotective effects demonstrated in rat models of Alzheimer's disease and age-related Parkinson's. There is a noted reduction in markers of oxidative stress in human animal models, namely by increasing total antioxidant capacity, glutathione levels, superoxide dismutase, glutathione peroxidase, glutathione reductase activities, and lowering blood markers of protein carbonyl and MDA levels. As previously mentioned, melatonin is generally supplemented within a range of 0.5 to 10 milligrams and is generally considered safe in doses up to 10 milligrams for 6 months. Although the 2020 systematic review and meta-analysis listed earlier showed that in patients under 35, there was a considerable effect on total antioxidant capacity by using 20 milligrams or more per day of melatonin. So if you're under 35, that's something to consider. There you have it. If you watched this far, you might be curious about what's coming up next on the Rise Above Health channel. Coming soon, I'm investigating the human gut microbiome and plan to demo a Viome microbiome test kit to gain a sense of my current gut balance. Viome is a very popular microbiome testing company that appears to use a method called metatranscriptomics to analyze the gut microbiome. More on that next time. Until then, I gotta go take a break because I've been doing a lot of talking. Take it easy, guys, and I'll see you on the next one.